next guest is coming from Champalimaud Foundation in Portugal that promotes and carries research in cutting-edge fields. Their mission is to stimulate discoveries that benefit people in areas of neuroscience and cancer. And both research and programs, clinical services, are carried out in Champalimaud. One of the examples is that they are involved in a program to find blindness, promote wealth and health and well-being. Anabel is a Ph.D. student at Champalimaud, and the study that she will present today, damage-responsive neuro-glial clusters coordinate the recruitment of dormant neural stem cells in Drosophila, is an important study needed to understand tissue repair involvement of stem cells that is crucial in this case of tissue repairs. So with this short intro, I will leave the floor entirely to Anabel.
Thank you. Hello, welcome. Thank you for inviting me to this meeting. So I'm going to share my screen. Can you see it?
Yes, we can see it. Yes.
Yeah. Okay. Good. Okay. So, as I told everybody, I'm a PhD student and at the Stem Cells and Regeneration Lab, and I'm going to tell you about the work that I've been doing during my PhD that's entitled Damage-Responsive Neuro-glial Clusters Coordinate the Recruitment of Dormant Neuroprogenitors in Drosophila. A very strong stimulus that dramatically changes the tissue environment locally and systemically is an injury. It can stimulate several forms of plasticity and initiate specific regeneration programs, which will then restore homeostasis. So regeneration is a long recognized phenomenon. It goes through from Greek mythology on the story of Prometheus, who had his liver plucked out by an eagle only to have it regenerate each day, or a superhero like Wolverine that has the ability to regenerate. So stem cells are important not only to maintain homeostasis, but also for regeneration upon injury. And adult stem cells are undifferentiated cells, which are found in most tissues throughout the human body. They are vital to renew and repair adult organs. They can self-renew and have the capacity to divide and give rise to daughter cells that will then differentiate into a tissue-specific cell type. There are different types of stem cells. There are ones required for homeostasis and others required for repair. The ones required for homeostasis are cycling stem cells, which are constantly cycled and are important for tissue maintenance. For example, there are tissues that constantly lose cells due to physiological turnover, like the epithelia of the intestine and the stomach and the skin. And there are quiescent stem cells, which are normally in G0, and they are able to enter the cell cycle upon injury. They are damage responsive. For example, there is the brain and the skeletal muscle. Sorry, can you see my cursor?
Yes.
Yes, okay, good. I was not sure. So in Drosophila, we can also find these two types of stem cells. We can find active adult stem cells in the ovary and the intestine, and we can find quiescent progenitor stem cells in the brain, the muscle, and the hindgut in the intestine. The mammalian brain contains mainly quiescent neural stem cells in two restricted areas, the subventricular zone and the subgranular zone in the dentate gyrus of the hippocampus. Although the quiescent neural stem cells have the intrinsic properties to mount a regenerative response to brain injury, the induced proliferation and associated neurogenesis is insufficient to support tissue repair in the lesion brain area. In Drosophila, until now we know that there are some neuroblast-like progenitor cells which still persist in the adult brain. And one characteristic of these cells is the expression of the S1 transcription factor called DEDPAN. This is important for neuroblast proliferation during development. They are normally scattered throughout the optic lobe through the medullary cortex. They are not in a niche. Upon an injury, by a stub lesion. But basically, we normally insert a thin metal filament through the eye of Drosophila. You can see it here. Stimulates proliferation and neurogenesis in the affected brain area, suggesting an ability to establish an extensive stem cell response zone in the adult brain. So upon an injury, the tissue environment changes locally and systemically, and once activated, Drosophila mic is upregulated, which is important for the transition from quiescent to the cycling state. These neuroblast-like progenitors are activated close to the stub lesion, and they start to proliferate. You can see here the mark with Ph.D, that is a marker for mitotic cells. And they give rise to new neurons. So, as I've mentioned, these dormant neuroprogenitors are activated by local brain injury. You can see here, 24 hours after injury. And they are found in clusters of cells that proliferated close to the lesion site. Upon injury, we can also observe some of the common initial responses to injury in an acute phase, like the activation of the GNK stress pathway. We also found upregulation of the engulfment receptor draper, which has been shown to promote clearance of neuronal debris in the adult brain on glial cells. Here you can see it colocalizes with glia. And we can also see apoptosis in the injury site. So a lot is known about the communication of the niches with stem cells and about homeostasis, but little is known on this distribution of stem cells and what drives this coordination. So our main goal was to see how is the stem cell activation zone defined. So which processes coordinate the recruitment of multiple and dispersed stem cells that are activated upon a localized injury. So these are the answers that I've tried to answer with my work. So in order to identify candidate signals that might be sensed by these quiescent neuroprogenitors, we studied injury-induced factors in the adult brain by genome-wide transcriptional profiling based on a microarray that was performed on injured versus non-injured brains 48 hours after damage. And from this array, a gene expression analysis was performed and the stringent cutoff was done, and 84 genes were differentially expressed, 76 upregulated and 8 downregulated. Gene ontology analysis show an enrichment of co-terms related to immune and stress responses. But most of the genes were associated with the extracellular region. So this analysis revealed a specific injury-induced secretome. You can see here. This is extremely interesting because secreted molecules could activate the stem cells locally. So the injury-induced genes included genes associated to innate immunity, like, for example, dipterosin, matchnicoin, edin. Also modulators of insulin like lymphostatin and IGDF, also cell adhesion molecules that matrix metalloproteinase 1, and a lipocalin-like transport molecule called SWIM. And among the most downregulated genes, we identify LSP2, which has been previously described as a repellent cue for axonal outgrowth at the neuromuscular junction. So then I went to validate in vivo the effects of these genes in proliferation. So under an ubiquitous acting GAL4 promoter and taking advantage of the temperature-sensitive repressor GAL-ATTS. So this GAL-ATTS inhibits the GAL4 at high temperatures. It's a repressor of the GAL4, and it's no longer repressed at high temperatures. Candidate genes to activate them. And the flies were kept at 18 degrees until adulthood. So we only wanted this effect on adult flies. And they were then placed at 29 to activate the RNAi. So the GAL4 is no longer repressed. And we did a lesion in the right optic lobe and waited 72 hours to allow them to proliferate. We then assessed the proliferative capacity using the phosphorescent tree. So this is the assay that I mostly use. So just be aware for the rest of the presentation. So I use LAX or expression of LAX-A and RNAi of MYC as controls. I told you before that MYC is upregulated upon injury. So, and these are all the genes that I validated from the microarray using RNA lines. You can see that several RNA lines increase proliferation, like the immune gene ataxin, or the metalloprotein is one, and others that suppress proliferation, like the all proteins, adenine, and the SWIM. We also validated the microarray results by independent quantitative expression analysis and confirm upregulation of SWIM and ninjurin A, that was also in the microarray, in accordance with these results. We also found upregulation of Drosophila MYC, which is in agreement with the results that I showed you before, that the MYC upregulation can drive proliferation in the optic. So based on these results, we then concluded that the microarray provides a faithful overview of major transcripts upregulated in response to brain damage. So from now on, I will focus on SWIM. SWIM is a secreted interacting molecule, which is a conserved protein which belongs to the life state, the extracellular transport of hydrophobic proteins, for example, wingless or winged mammals. And it facilitates the wingless diffusion throughout the extracellular matrix. So this is just a scheme of the configuration of the lipocalin. So they fold in a cup-shaped cavity and bind and conceal the lipid component of their target molecule. So you see here the target molecule. So this happens by covalent attachment of palmitic acid and palmitoleic acid. So this is a lipid-dependent manner. So SWIM binds to wingless with nanomolar affinity in a palmitate-dependent manner, which maintains its solubility and allows it then to be transported. So previous experiments have shown that SWIM transcripts are enriched in the larva brain, and it also tested the binding affinity to wingless for full-length SWIM and to its C-terminal fragments. And in both cases, it was higher than the high affinity binding domain of the wingless receptor, the Frizzle receptor. So then our next question was, is SWIM important in response to an injury? So by using this same assay, we have here two different RNAi lines for SWIM, and in both cases, it significantly reduced injury-induced cell division. We also knocked down levels of DALI-like. So this is an heparan sulfate proteoglycan that has been shown to act as a wingless transporter in developing the cell divisions. So this provides additional evidence that SWIM may act as a relevant transporter in the brain. So then, to study a complete loss of function in a highly specific manner, we generated a SWIM knockout by removing the entire SWIM coding sequence via CRISPR-Cas9. So the adult SWIM knockout flies did not show any defects. You have the control and here the knockout, so they were pretty normal. But however, when we challenged them with the acute brain injury by stab lesion, the SWIM knockout show a decrease in injury-induced proliferation. You have here an image, you have pretty much no pH3 compared with the control. So this is in line with the results from the RNAi. And This shows that swim is required for injury-induced proliferation, but dispensable for development. So then we went to verify if swim is actually produced in the injured brain. And for this, we created swim knock-in flies by integration of the target genes by a recombination of a TTP and TTB with a knockout. So where then we'll be able to monitor the expression of the fluorescently marked swim upon injury. So this line, secretion is induced. So swim was pretty much undetectable with cell type specific markers, shown that was induced in glial cells, allows infection in glial cells to drive proliferation. So by using the same assay of the proliferation,
I think we lost connections with
sorry.
Anabel.
Can you hear me?
Yeah, we can hear you.
Can you hear me now? Okay. Yes. So where did it stop?
When you talked about proliferation again.
When you talk about?
Proliferation. We are losing.
I talked about proliferation already a lot, so I don't know which part.
Let me show. Let me see your presentation.
Maybe here?
Wait.
Or no, more or less? You already saw this part.
Yeah.
Yes, so yeah. This part as well.
Yes, yes.
This part here.
Yes, we passed through that also, I think.
Okay.
Yeah, we passed through that.
Here.
A little bit forward. If you can. Anabel, can you hear us?
So maybe, so here.
Yes, here.
Sorry, I don't know why. Normally it's very stable, but today. Can you hear me?
Yes, we can hear you.
Okay, so I think it was from here or no?
Yes, yes, it was from
okay.
here.
Okay, cool. So I'll just continue. So to test swim function in glial cells to drive proliferation, so I used the same assay, but this time with a glial driver. I have knocked down swim expression using two different RNAi lines in adult glia and observe a drastic reduction of cell division. So this indicates that swim expression in glia is essential to promote proliferation following an injury. So we also evaluated the type of glia which expresses swim by using a glial subset specific driver. And we found that swim is expressed in in-sheeting glia. So this glia is known for clearance of debris and to act as a barrier to interrupt the neuropil. So secretion. So since there are evidence that swim can transport wingless, we first asked if swim response correlated with increased wingless pathway activity. So we therefore assessed the promoter activity of the flywing gene in injured brains, express the wingless-driven GFP, and we found consistent activation of the wingless promoter, so the GFP positive cells, in the discrete set of cells in the injured optic lobe 48 hours after injury, compared to non-injured brain. You see here the quantification, and this was no longer distinguishable after 1 week. So wingless is induced in the injured brain. but in which cells is Wnt induced? So since the Wnt signals released from glial cells have been described to induce proliferation in the context of tissue regeneration, we checked if the Wnt expressing cells correlated with apoptotic markers, such as the activated effector caspase DCP1. But this was not the case, as you can see here. And then we stained the injured range for cell type specific markers and found that Wnt was not induced in glial cells. Here you see there's no localization,
Anabel,
but rather yes.
Sorry.
Can you hear me?
Yes, I can hear you. I will kindly ask you to turn down the camera, and
yes, of course,
because it might work better.
Okay.
Thank you.
Okay. It's there. Okay. So we stained the injury range for cell type specific markers and found that Wnt was not induced in glial cells, as you can see here by the ripple staining, but rather occurred in a restricted number of LIV positive neurons located at the lesion brand area. You can see here, go localize it. So then we observe at tissue scale Wnt expressing neurons and swim area expressing glia, and they could be found in proximity, forming these multicellular neuro-glial clusters with different sizes and composition. So this is how we think that the environment is within the injured brain. So we call them damage responsive neuro-glial clusters. So the proliferative response is governed by these neuro-glial clusters. So then, to explore the potential interaction of swim with Wnt, we assess Wnt distribution in injured versus non-injured brains with a Wnt antibody. So you have here the two conditions. So in the absence of damage, so completely non-injured brains, Wnt staining was restricted to a few cells in the adult optic lobe. At 48 hours after damage, you see that were already present in several cells in the injured optic lobe. So to check if the surge of swim expression affects the brain injury is relevant for the Wnt distribution in the injured brain area, we suppress swim induction in adult flies with the RIF swim and monitor Wnt protein follow injury. And you see here, just by looking at the image to the control, and you see here the quantification. And the same was observed with the swim knockout flies. You see a lot less Wnt cells compared with the control. So swim promotes Wnt mobilization in the injured brain area. So then to check if the Wnt signal transduction is required cell autonomously in quiescent neuroprogenitors to enter a proliferative state, we set out to generate a deadpan-dependent driver, which can drive strong expression of target sequences specifically in quiescent neuroprogenitors. So then to block the development deadpan expression, we activated this deadpan GAL4 exclusively in adult flies, so with the GAL-ATTS, and combine it with G-trace cassettes to monitor real-time expression of deadpan with UAS RFP, or cells that have previously expressed deadpan with GFP. So using this approach, we confirmed deadpan expression, these red cells, in undifferentiated cells, so they are either LIV or repo, shortly after injury. So this was 24 hours after injury. Just for the following thing, I will quickly guide you through the canonical Wnt pathway. So in the presence of the Wnt ligand, Wnt binds to frizzle and arrow, triggering the translocation of the destruction complex to the inner plasma membrane, and it gets inactive. So without this phosphorylation, armadillo concentration rises and enters the nucleus, where it binds to the nuclear transcription factor TCF to regulate Wnt target genes. So then we use the and proliferative assay to manipulate wingless signaling, but this time using the deadpan driver. So as we expected, when we RNAi MYC in adult neuroprogenitors, it abolished the injury-induced proliferation. When we inhibit an important wingless receptor, frizzle, I talk here, with a dominant negative version of it, it also strongly reduces proliferation compared to control flies. In contrast, when we overexpress wingless in progenitors, it efficiently stimulated proliferation even in the absence of injury. And the same was observed when we overexpress MYC. So we can conclude that canonical wingless and Drosophila MYC are required for the proliferative response in quiescent neuroprogenitors. So, but what controls swim induction upstream? So we know that the brain injury leads to several pathophysiological responses, including oxidative stress, apoptosis, and hypoxia. So, and literature has given some hints to a relationship between hypoxia and swim. So we went to check. So hypoxia inducible factor 1 alpha, if1 alpha, is a transcription factor which responds to a decrease in available oxygen in the cellular environment. So if1 alpha stabilization and binding to hypoxia responsive elements to regulate gene transcription is strongly conserved in mammals and Drosophila. So we therefore assess swim upregulation by real-time PCR in SEMA no mutant. So SEMA is the name in Drosophila for if1 alpha. So these flies are deficient for the gene if1 alpha, and the swim transcripts were no longer upregulated in these flies. So swim is dependent on if1 alpha. But does an injury actually causes hypoxia? So for this, we use an in vivo sensor which responds to cell oxygenation. So it's a fluorescent timer protein. It shows the levels of oxygen in different tissues. And so at high oxygen levels, it turns into a red fluorescent isoform, so it's in normoxia, and at low oxygen levels to a green and blue isoform, so it's in hypoxia. And we use it in combination with the HOC L4 to only induce it in adults. Here you can see a brain with no induction. It has a little background expression, but not so much. And with induction, and so upon HOC, in a homeostatic condition, so no injury. When we injure the right optic lobe, we can see increased levels of green and blue fluorescent isoform. Here in the here you can see it better compared to the is activated upon injury. So swim function is likely modulated by injury-induced hypoxia. So then to establish if hypoxia alone would be sufficient to trigger swim expression without the injury, we expose the swim M-cherry flies to hypoxia by using an hypoxic chamber. So this is a chamber, this is the gas mixed. So we confirm that both in the larvae and in the adult brain, hypoxia was sufficient to induce M-cherry expression compared to the normoxia conditions. So swim induction is regulated by hypoxia in an if1 alpha dependent manner. We then turn to mouse models of traumatic and ischemic brain injury to address whether these hypoxia-dependent injury sensor mechanisms that control swim expression is conserved in mammals. And the function of the mammalian homologue of swim, that's called lipocalin-7, is unknown. In a collaboration with the Basque Center Institute of Neuroscience, which has a model of traumatic brain injury that was caused by controlled cortical impact, led to induction of lipocalin-7, similar to our observations of acute brain injury in Drosophila. So under homeostatic condition, lipocalin-7 was not detectable, not even in sham conditions. And in contrast, it was clearly upregulated 3 days after a traumatic brain injury, and it did overlap with GFAP positive astrocytes of the dendrite gyrus, but not with nested GFP labeled neuron cells. In a different collaboration with the group at Chapalimau, the immunophysiology lab, they had a stroke model, and it was also able to see induction of lipocalin-7 in the stroke side. So basically they did a transient middle cerebral artery occlusion in one hemisphere, and the other side is attached to the contralateral. And you see here the contralateral pretty much don't have signal, and this again colocalized with glial cells in the hippocampus. So injury to stream induction in glial cells is conserved in the mammalian brain. Just to finalize, our model is that the traumatic brain injury causes transient hypoxia. There is secretion of restricted amounts of wingless ligands from neurons. Glia responds to transient hypoxia by upregulation of SIM in an IF and alpha dependent manner. SWIM binds to hydrophobic wingless and promotes its distribution in the injured brain area, promoting a transitory activated environment for circulating neuroprogenitors, and wingless signal in quiescent neuroprogenitors and downstream upregulation of MIC promotes proliferation and eventual group. This is Krista. This is for those of you who don't know, this is Chapalimau Foundation in Lisbon. And these are the people that were involved in the project that were very, very important for me to finish it. And we are also hiring postdocs if any of you is interested. So thank you. So if you have any questions, please feel free.
Thank you, Anabel. We have to wait a little bit to see if there are questions. Please use Q&A function of Zoom to write questions.
Actually, I have one question. How can you verify that there's a similar mechanisms in humans?
How can you verify? So there are, normally it's done by post-mortem tissues that they use, like by stainings, in vivo just by cell culture, for example. But yeah, so for example, as I show you, there's this, for example, antibody for the lipocalin that can stain the winged signal pathway is a very conserved pathway in the several species, and it's known for acting in several things. So there is ways. Cell culture probably a way of easily testing this. Getting access to slices of human brain will be a little bit tougher, but yeah, probably the cell cultures.
I also want to ask, how can these findings be translated into therapies?
So, for example, stimulating the transport of SWIM, or the secretion of SWIM, to allow to transport wingless, to then increase the pool of stem cells to improve regeneration, because this is the critical point of regeneration, is that it's not enough for repair. So there needs to be strategies to improve the pool of stem cells to then be able to differentiate into the needed cell types. So this will be a way to do it. So there are already studies, not with SWIM, but with wingless, that use wingless to try to improve this pool of stem cells and regeneration. This is very, very known, even in cancer, for other types of things. So this is a very well described factor.
Thank you.
You're welcome. Any more questions?
No more questions. Thank you, Anabel, for coming.
You're welcome. Thank you for inviting.